A blood cell, also called a hematocyte, is a cell produced by hematopoiesis and normally found in blood. In mammals, these fall into three general categories: red blood cells are erythrocytes, white blood cells are leukocytes, platelets are thrombocytes. Together, these three kinds of blood cells add up to a total 45% of the blood tissue by volume, with the remaining 55% of the volume composed of plasma, the liquid component of blood. This volume percentage of cells to total volume is called hematocrit, determined by centrifuge or flow cytometry. Hemoglobin is an iron-containing protein that facilitates transportation of oxygen from the lungs to tissues and carbon dioxide from tissues to the lungs. Red blood cells. Red blood cells primarily carry oxygen and collect carbon dioxide through the use of hemoglobin, and have a lifetime of about 120 days. In the process of being formed, they go through being a monopotent stem cell. They have the job alongside the white blood cells of protecting the healthy cells. White blood cells. White blood cells are cells of the immune system involved in defending the body against both infectious disease and foreign materials. 5 different and diverse types of leukocytes exist, but they are all produced and derived from a multipotent cell in the bone marrow known as a hematopoietic stem cell. They live for about 3 to 4 days in the average human body. Leukocytes are found throughout the body, including the blood and lymphatic system. Platelets. Platelets, or thrombocytes or yellow blood cells, are very small, irregularly shaped clear cell fragments, 2-3 micrometer in diameter, which derive from fragmentation of precursor megakaryocytes. The average lifespan of a platelet is normally just 5 to 9 days. Platelets are a natural source of growth factors. They circulate in the blood of mammals and are involved in hemostasis, leading to the formation of blood clots. Platelets release thread-like fibers to form these clots. If the number of platelets is too low, excessive bleeding can occur. However, if the number of platelets is too high, blood clots can form thrombosis, which may obstruct blood vessels and result in such events as a stroke, myocardial infarction, pulmonary embolism, or blockage of blood vessels to other parts of the body, such as the extremities of the arms or legs. An abnormality or disease of the platelets is called a thrombocytopathy, which can be either a low number of platelets, a decrease in function of platelets, or an increase in the number of platelets. There are disorders that reduce the number of platelets, such as heparin-induced thrombocytopenia or thrombotic thrombocytopenic purpura, that typically cause thromboses, or clots, instead of bleeding. Platelets release a multitude of growth factors including platelet-derived growth factor, a potent chemotactic agent, and TGF-beta, which stimulates the deposition of extracellular matrix. Both of these growth factors have been shown to play a significant role in the repair and regeneration of connective tissues. Other healing-associated growth factors produced by platelets include basic fibroblast growth factor, insulin-like growth factor 1, platelet-derived epidermal growth factor, and vascular endothelial growth factor. Local application of these factors in increased concentrations through platelet-rich plasma has been used as an adjunct to wound healing for several decades. Complete blood count. A complete blood count is a test panel requested by a doctor or other medical professional that gives information about the cells in a patient's blood. A scientist or lab technician performs the requested testing and provides the requesting medical professional with the results of the CBC. In the past, counting the cells in a patient's blood was performed manually, by viewing a slide prepared with a sample of the patient's blood under a microscope. Today, this process is generally automated by use of an automated analyzer, with only approximately 10 to 20% of samples now being examined manually. Abnormally high or low counts may indicate the presence of many forms of disease, and hence blood counts are amongst the most commonly performed blood tests in medicine, as they can provide an overview of a patient's general health status. Discovery. In 1658, Dutch naturalist Jan Swammerdam was the first person to observe red blood cells under a microscope, and in 1695, microscopist Antony van Leeuwenhoek, also Dutch, was the first to draw an illustration of red corpuscles, as they were called. No further blood cells were discovered until 1842 when French physician Alfred Donner Copyright discovered platelets. The following year leukocytes were first observed by Gabriel Andral, a French professor of medicine, and William Addison, a British physician, simultaneously. Both men believed that both red and white cells were altered in disease. With these discoveries, hematology, a new field of medicine, was established. Even though agents for staining tissues and cells were available, almost no advances were made in knowledge about the morphology of blood cells until 1879, when Paul Ehrlich published his technique for staining blood films and his method for differential blood cell counting. References. External links.